I'd like to thank again the organizers for the Molecular Frontiers Foundation. And I think it's a wonderful opportunity to get high school students in Japan and elsewhere enthused and interested in science. I'll start by telling a little story. When I was a high school senior in Phoenix, Arizona, we had a professor from Caltech come out to give a talk on black holes. The guy's name was Kip Thorne. Kip just won the Nobel Prize in Physics for the LIGO project and the discovery of gravity waves. But because of Kip Thorne traveling all the way to Phoenix, Arizona, and talking to a group about this size of high school students, I applied to Caltech. It really changed my mind. Here was a young, dynamic professor talking about huge energy sources. It was fantastic. I've never regretted since then going into the sciences, because everything I do and see is more fun than ever. So today, I would like to leave you with a message that if you find something that grabs your mind and takes your interest, don't let it go. Follow it wherever it goes. And I'll show you what happened to me when I was a freshman at Caltech back in 1971. And first we'll acknowledge it's about magnetism. It's about biology. When I was a high school student, I wanted to be a physicist, I wanted to be a geologist, and I wanted to be a biologist. And so when I got to Caltech, I couldn't choose. And so I did everything. I wound up as how do you do it? A geophysical neurobiologist, an earth historian, I don't know what to call myself. But first, I would also like to say that as you grow in science, you have to thank the people that give you money. And a lot of organizations have given my laboratory money. Almost none of them knew what we were doing. The one that did was the Human Frontiers Science Program. Is there a laser? Yeah. Human Frontiers Science Program, actually, a few years ago, did something very crazy. They gave us money to look for human magnetic sense. Very crazy. So what I would like to do is go tell you about the magnetic bacteria first. They're the clearest example of a magnetic organism. So the magnetic biosphere, I really mean magnetic biosphere. Watashi tashi, Zimbu life. It's magnetic. And using it, I mean, not just attractive, but using it. Okay. And then I'll talk about how we know this from eukaryotic protists to animals. And at the very end, I'm going to tell you something that's not yet published, but it's blowing your brain, blowing my brain anyway, that humans are animals. Humans are animals? We are also animals. And it turns out we have a magnetic sense like many animals. It's been hidden. It's not in the conscious. And you might call it a sixth sense, but Aristotle didn't count a few other ones, so it's like the 10th or 11th sense. Okay, so first, not just magnetic bacteria, but I'm going to show you how I got involved with this. Can we turn the lights down just a little bit? Denki ga choto okimasu. All right, yeah, so when I got to Caltech, the faculty were as confused as I was. I wanted to be a geophysical biologist, and the closest person they could give me as an academic advisor was a paleontologist. And this is Heinz Lowenstamm. And as soon as he heard I wanted to be a geophysical biologist, he said, "Oh, I want to show you something." And what he showed me were these small mollusks, the chitons. And it turns out they live in the intertidal zone, and they eat rock. Really, they eat rock, all right? And because there are algae in it, but they needed hard teeth. If they didn't have hard teeth, they couldn't do it. So the next slide, I will show you what grabbed my brain in 1971 and changed my life forever. This. Meet it. These are the teeth. Hades. The teeth are made of a mineral that is magnetic, magnetite. And in fact, I have the teeth right here. Maybe turn the light up a little bit, yeah. You can pass this around. It's a little magnet, and you can just, if it doesn't move, tap it. It's a little sticky. Yeah. Anyway, but the point is, the teeth are magnetic. I'll do that again. You can actually see it. Yeah, the teeth are magnetic, and you can pull them around. And actually, as an undergraduate, I studied these with Heinz doing electron microscopy. These are magnetite teeth, very clearly made by biology. Now, magnetite is a metal, okay? It conducts electricity. It's highly magnetic. It's strange. That grabbed my mind. And it's biological. The crystals are here. They're inside protein, actually collagen fibers. And it is the hardest material that organisms make. So, well, it turns out about 3 years after that, a graduate student at Woods Hole discovered another animal organism that makes magnetite. And these are the magnetotactic bacteria. And in fact, as I'll show you a bit, we're going to do an exercise with our student group extracting these bacteria from mud samples that we collected down the road here from Tokyo Tech. So anyway, if you put bar magnets here, you'll notice the white powder here, those are billions of magnetic cells that swim toward the magnet. And you can see it. You turn the magnet. All right, you turn the magnet, and billions of bacteria move with the magnet. I mean, I saw that and I said, oh, well, it's not just the chitons, it has to be these other ones. So this is a couple weeks ago with the student assistants here. We went to a little pond and pulled out mud samples and do the same A, B, C, D, E. We have these samples in the lab for tomorrow morning. We'll put magnets next to them and go in and extract the magnetic bacteria. We'll be able to see that tomorrow. So they're fun, but so what? Remember, the theme here is something grabs your mind, run with it. And I've been running with this for almost 40 years now. And we now know, of course, that the magnetite are in chains, but we know a lot more. It's naturally magnetic. You don't have to magnetize it spontaneously. It's a ferromagnetic material. The Bohr magnetines line up, and it does its thing. They're inside the cells. This is the same way you make teeth or bones. It's a biomineral, genetically controlled. So the DNA comes in. And totemo chisai desu. Very small crystals. 20 to, actually about 30 to 100 nanometers in size. But biologically controlled. Well, actually, the crystals are held in place by a cytoskeleton. That was actually the first discovery of a bacterial cytoskeleton. And my wife made that discovery with TEM work. Yeah, that was Atsuko Kobayashi. That was her thing. Anyway, if you look at these bacteria, we know what they use magnetism for. They want to go down. The magnetic field allows the bacteria to swim down. Most bacteria do a random walk. These bacteria know where they're going. In fact, I discovered the south-seeking bacteria a few years later. Blakemore did the first one in North America. And on the magnetic equator, they go both directions. So this is kind of neat. They use the field for up and down Magnetic field is horizontal on the equator. Otherwise, they do a random walk, so they're fun. Now, I love this one because remember at Tokyo Tech, one of the major scientific and engineering advances was magnetic recording particles, the ferrite industry. There's a whole museum here right up the road showing Professor Takei, the TDK, anyway. But what that means, it turns out if you have a little crystal of magnetite, it can be magnetized this way or that way. And a little magnetic pulse can flip the direction between the two. So if you do that to our friendly north-seeking bacterium, it'll roll over and swim south. In fact, we can do that. And if you do the pulse the other direction, it won't change. So it's a directed pulse. So that is the fingerprint of ferromagnetic effects. I'm going to show you this experiment 3 times in this lecture. So again, the ferrite industry, this is Tokyo Tech up the road. If you are on campus, visit that museum. It's fantastic. Is it open on Sunday? I don't know. Gomenasai. Anyway, it's wonderful. But what we can do is take a little capacitive unit and make a magnetic pulse, and that will be enough. So I'm going to show a movie of the magnetic bacteria, well, actually in 2 slides. But I'm controlling the pulse so that half of the bacteria get remagnetized. And this is an example of what that does. It takes a compass and remagnetizes it, okay? You'll see this again here in a bit. Anyway, so here's the magnetic bacteria. This is what our students will see tomorrow. We make the magnetic bacteria swim this way, turn this way, turn that way. I can control billions of bacteria with magnetism. And I'm going to make them collect there. Then I'm going to come here. I'm going to turn them around. Turn around. Thank you. And then bang. I hit it with a pulse. Now I have 2 waves. Okay? Half of them have been remagnetized. Biomagnetism. That's a magnetic biosphere. Very clear. Okay? So, question. Oh, yeah. I'll show you. Turns out we now know from a paper we published last January that this ability most likely evolved very early in the history of life, probably around 3.4, 3.2 billion, Sunjian Okunin, more or less. And the reason we can distinguish that is because of a gene inversion in the part of the DNA on the chromosome that builds the magnetite. There's an inversion in the nitrospirae that has persisted in that phylum since that phylum separated. And it turns out that split was back here phylogenetically, and that's that gene order split. And so magnetotaxis has to predate all of this. And it's not lateral gene transfer that we can rule out. It's very elegant. And in fact, the magnetosome genes follow the same lineages as the ribosomes. So it's pretty clear, not moved around. But if you put that on a timeline with a great oxygenation event, that puts it back here, 3.2 billion. Now, Sunjini Okunin Maides for the magnetic bacteria. So magnetotactic bacteria are there. Very, very ancient forms of life. So if we make a tree of life now, our data from this slide roughly is here, near the base of the bacterial domain. And then magnetite biomineralization, we know, is in many bacterial groups. It's easy to lose, but it turns out the ancestors of most bacteria had to be magnetotactic by the way that evolution worked. You know, they lose it, but the ancestors had it. And then something happened with the mitochondria, formed eukaryotes, and things went up here. And I'll show here, we have very, very similar machinery and magnets, all the way up to mammals and beyond. But the question is, did magnetite, how much earlier? So here's some fun. Is there any evidence for magnetic bacteria earlier than that? Anybody? Yes? Of course, the students who read the papers would know. And the answer is yes. What we were able to show is that this meteorite, which is Martian in origin, it formed about 4.2 Yonjuni Okunin billion years ago, 4.2 billion, came to Earth without heating even to 40 degrees. That's another whole study out of my lab from the magnetism. But that is the meteorite that caused so much controversy back in 1996 when David McKay and his team argued for Martian life. And the cartoons were wonderful. My God, the scientist here says, well, we think we might possibly have 4 lines of evidence from this Martian rock that might possibly imply there was life on Mars. And of course, everybody goes crazy. Turns out, of those 4 lines of evidence, actually, all of them are still lines of evidence. There are funny structures that look biological. There are organic materials there that came from them. There are disequilibrium iron minerals. But the last one they suggested has never found any other interpretation other than biology. If you go to this meteorite, there are carbonate pancakes, very thin film of limestone, which contain magnetite. If you go in and look at those magnetite crystals, they have a morphology that the ferrite industry, this invention of Tokyo Tech back in the 1930s, the ferrite industry has never been able to make even a single crystal of magnetite of this size, shape, chemical purity, structure. No one has made it without biology. And the bacteria cheat. They make a box and then fill it in. And it gets rid of diffusion problems and chemistry problems. But really, you can't distinguish these things from Mars from bacterial precipitants. So it's still a very viable idea that we might actually have had magnetic bacteria on Mars. So interesting. Well, let's move on. The rest of Earth's biosphere is also magnetic. And the previous, well, I mean, I showed you the chitin teeth. The chitin teeth are moving around somewhere, right? Yeah, pass it around, look at it. But it turns out there are also other eukaryotes, not the mollusks, that make magnetite. There are groups of very primitive protists, single-celled eukaryotes, not bacteria, that also make magnetosomes. We might see some tomorrow. We should look. They're very big cells that come into the magnetic field, and they turn the magnetic field over, and they go the other way. We always try to find them because they're a mystery. There'd be a great PhD career for somebody. Anyway, but they're there. We also, from the fossil record, know that there are some monsters out there. This is a single magnetite crystal, 55 million years old, from what we call the Paleocene-Eocene thermal maximum. The sediments load up with magnetic crystals, with magnetic fossils. And we occasionally see magnetite spearheads. This is like a Viking spear or something, you go point things. We know roughly what they look like. They occur in an assemblage of about 15 micron ball. So we call it the Death Star. We don't know what it's doing, but it's clearly biological. The crystals are sculpted exactly the same. So those are examples of the magnetic biosphere, but we also know that many animals use the magnetic field to navigate. And so it's not just the bacteria, not just the protists. It's a well-known and well-studied sensory system in things. It's not the 6th sense. I mean, Aristotle enumerated 5 senses. He forgot about gravity. He forgot about proprioreception, inertia. He forgot about time and temperature. I mean, Aristotle missed a few. But magnetism, we now know, is in everything from mollusks to arthropods to fish, reptiles, birds, and mammals. And I'll show you a few examples of this, just the ones that kind of grab my mind. Honeybees, Mitsbachi, are lab rats. I spent 10 years training honeybees in my lab because you could train them to magnetic anomalies. And then there were about a dozen experiments on bees. Some are very simple. If you take a new swarm of honeybees, 6,000 honeybees, put them in a dark barrel, they have to make new honeycomb. Turns out the direction that that new honeycomb is made in is the same magnetic direction of the parent hive that the swarm came from. And in this case, this is Martin Lindauer and Hermann Martin in Würzburg. It was within 1 degree and then precisely there, the new honeycomb. How do they, see, 6,000 bees have to agree on a direction. If there's no other cue, they use magnetism. The demonstration that it's magnetic, if you put a big magnet on top of the barrel, that's what you get. A brick doesn't do it, a magnet does that. They get lost. It's a very simple, I mean, this is like the magnetic bacteria, but for bees. Okay. In birds, there are, again, dozens of studies. If you release birds on a magnetic anomaly, in bright sunlight, they get confused. If you release them away from the magnetic anomaly, they figure out home, and they fly home. These are Charles Walcott's data. That's Walcott's bird. Whales, Kojiro, turns out when you're in the ocean, you need to navigate. And as a geologist and a geophysicist, we all know that the magnetic field over the ocean is a very regular map. If you look at magnetic intensity, high magnetism and low magnetism, we have stripes. There's parallel symmetry. We now know that the magnetic field is symmetrical around spreading axes. The magnetic seafloor forms at the spreading ridges. As it moves away, the magnetic field reverses, and you end up with patterns of stripes. Well, turns out we have very good evidence that the magnetic stripe patterns are the map that whales use at sea. And it turns out whales make mistakes occasionally, and where they make mistakes turn out to be magnetic anomalies. And in fact, this is a picture of the magnetic anomaly map of the east coast of the United States. You can see the east coast magnetic anomaly coming here. The red dots are places where groups of whales alive tried to strand. Some mistake is being made. We don't know why they strand, but we know where. And it turns out where they strand tends to be where these magnetic minima cross the coastline. And you can see it. Here's a plot of the magnetic field from south to north. These are the number of whale strandings at places. Particularly the big whale strandings all seem to occur where the magnetic minima crosses the coastline. So, but that's a good strategy unless you're near a continent. But most of the whales that do strand are species that usually don't come near continents. They get confused. And when they do, they try to navigate, and they follow the magnetic field. So this then leads to the search for the magnetic receptors. How do they do it? Okay, and actually, because of my work on the chitin teeth and the magnetic bacteria, when I got to Princeton as a graduate student, there was a group in biology led by Jim Gould, whose students were pulling their hair out because they had bees that were replicating magnetic experiments. And so I kind of said, oh, maybe there's a magnet in the bees. And being a geophysicist and having studied rock magnetism with Gene Shoemaker, the Shoemaker-Levy comet, I said, oh, maybe there's magnetite there, just the same idea. And it turns out there is magnetite, and this is just a cartoon by Charles Schultz. You know you've made it when a very famous cartoon author writes a cartoon strip about your PhD thesis. Okay. So this is fun. So, but there are actually 3 possible mechanisms for magnetic reception. The easiest and simplest is electrical induction. Sharks, we know, when they swim through the magnetic field, the induced electric currents can be detected by the shark electric sense. So that's a reasonable idea. It works for sharks. Other people have proposed, Klaus Schulten, for example, and Ritz, that maybe it's a quantum mechanical effect. If you have a photon absorbed by a molecule that then creates a free radical, the hyperfine split of the geomagnetic field might be enough to cause a chemical reaction difference. The triplet to singlet decay is inhibited. And they have proposed that birds may have in the retina, particularly a chemical called cryptochrome, that might be a free radical mediated quantum compass. I'm actually going to show this is not correct in a bit. And the reason that I disagree with this is that a quantum compass cannot detect the polarity of the magnetic field. It's this Hamiltonian problem. It's b squared, and it's the energy, and it doesn't matter on the direction. Well, the problem is birds seem to follow that rule. If you have a bird that wants to go north, and you flip the horizontal component, the bird will then fly south. But if you then flip both components, the bird still flies that way. Okay? It seems to be polar insensitive. It can't tell. It uses the angle the field is dipping rather than the actual component of it. Now, when I saw that, I said, well, you know, that's probably the brain processing it for some reason. It doesn't mean the sense organ does that. But because it agrees with the quantum compass, all the chemists think, oh, no, it's a quantum compass. Yeah. Well, the third idea is, again, magnetite, my favorite material. And in fact, 30 years ago now, we were able to extract gently magnetosome chains, magnetite crystals, from the olfactory tissue of salmon and other fish and many other animals. The salmon who had the cleanest materials because it worked well. We also know that even human tissues typically have magnetosomes like this. This is my wife's, Atsuko Kobashi's, TEM images of the human brain magnetite we published about 25 years ago. So the magnetite is there. I'm going to now show you an experiment that proves that mammals use magnetite. Here's the experiment. We take a little brown bat. This is Dick Holland, who was a postdoc at Princeton about 15 years ago. And we take the bats and move them north of Princeton and release them. We can see which way they go. But we take my little pulse magnetizer, again, that makes the little pulse. And we can hit a single pulse on the bat and see what happens. So here's the paper we published about 10 years ago. The control bats flew south towards Princeton very nicely. The parallel pulse, remember I showed you that you could pulse it, and if it's the same direction, it does not change. If you pulse it the other way, it rotates. Well, parallel pulsed has almost no effect. No significance. However, when you pulse it the other way, they're totally confused. Around. We don't understand exactly. I was thinking they should all go completely the other direction. But it turns out it's not so simple. If it were just one cell, it might go the other way. But we now think there may be actually many cells directed in different orientations that are communicating to the brain for that.